Hey Saturton, this is Mr. Dubik, and in this YouTube video we're going to be learning about the Krebs cycle and the electron transport chain, which are the last two stages of cell respiration. Yesterday we talked about glycolysis and the prep stage of cell respiration. So on this slide, what I would like to do is just recap what we learned about yesterday. Yesterday we learned about the first two stages of cell respiration, which is glycolysis and the prep stage. So remember, in glycolysis, one single glucose molecule from photosynthesis is broken down into two molecules of pyruvate. Also, remember that we produce a total of 4 ATP molecules, but we use 2 ATP molecules to split this 6-carbon molecule into 2 3-carbons. So we only net 2 ATP molecules, and then also because bonds are being broken, electrons are going to be released. So as a result, NAD plus is going to pick up those electrons and create NADH, which carry electrons and hydrogens to the electron transport chain. Alright, so at the end of glycolysis, we have 2 molecules of pyruvate. Now these 2 molecules of pyruvate are going to then enter the mitochondria. Okay, and remember that glycolysis occurs in the cytoplasm of the cell. So now these 2 molecules of pyruvate are going to enter the mitochondria into the prep reaction of cell respiration. And remember, in the prep reaction of cell respiration, these 2 molecules of pyruvate are going to bind with coenzyme A to form acetyl-CoA. Okay, and as a result of coenzyme A binding to pyruvate, you're gonna lose a carboxyl group, which is going to be released as carbon dioxide. That's what we exhale out of our mouths. And as a result, we form acetyl-CoA, and acetyl-CoA is now going to be used in the Krebs cycle to essentially energize it and start this cycle. So now what we're going to talk about is we're going to talk about what actually happens in the Krebs cycle, what is produced in the Krebs cycle that is so important for the electron transport chain, and then we're going to talk about the electron transport chain and what its main job is in cell respiration. So here we go. Okay, so the third stage in cell respiration is the Krebs cycle. And just essentially an overview of what the Krebs cycle is. It's an energy extracting process that breaks down acetyl-CoA to form the electron carriers NAD, NADH, and FAD, FADH2, which fuels the production of ATP. So essentially the Krebs cycle, the whole purpose of the Krebs cycle is to convert acetyl-CoA into NADH and FADH2, which are electron carriers. And essentially those electrons and hydrogens are going to be used in the electron transport chain to make ATP. Okay, this is an aerobic process, so in order for this process to occur, in order for the Krebs cycle to run and work, oxygen must be present in the environment. Where does the Krebs cycle occur? It occurs in the mitochondrial matrix. And again, like I said before, it's a series of energy extracting reactions. And essentially the purpose of the Krebs cycle is it's going to produce NAD and FAD, which carry electrons and hydrogen ions needed in the electron transport chain to make ATP. Okay, so what we're going to do on this slide is we're just going to talk a little bit about the reactants and the products, go over this simple diagram, and then move on to the electron transport chain. So the reactants, what is needed for the Krebs cycle? Well, we talked about in the prep stage that pyruvate cannot enter the Krebs cycle. So what do we need to do? We need to take that pyruvic acid, bond it with coenzyme A to form acetyl-CoA. So acetyl-CoA is going to initiate the Krebs cycle. It's going to get it running. Also, we have NAD+, FAD, and ADP. Okay, so these are going to be our reactants. So acetyl-CoA is going to initiate this process, and as acetyl-CoA, and now I'm going to talk about the diagram, as acetyl-CoA is being broken down through the Krebs cycle, you can see that this molecule, acetyl-CoA, if you follow it, it's being broken down. Okay, it's changing throughout the Krebs cycle. And as you see that, what's happening is bonds are being broken. And as bonds are being broken, okay, electrons are being released. And who's going to come in to pick up those electrons? NAD plus and FADH2. And as a result, now we're forming and creating these high energy electron carrying molecules that essentially are then going to go to the electron transport chain to make ATP. So what are the reactants in Krebs cycle? 2-acetyl-CoA, NAD+, FAD, and ADP. What are the products? So what are we essentially producing in the Krebs cycle that is so important? Well, first we're producing carbon dioxide. So again, this is the other 2/3 that we exhale, okay? And that just comes from right here. So a carboxyl group, another carboxyl group is going to be released in the form of carbon dioxide. We're also forming our 2 energy carrier molecules, NAD and FAD. Okay, so you see that when citrate is broken down here, okay, electrons are going to be released, and then NAD+ is going to pick up those electrons. It's going to be reduced, and it's then going to pick up a hydrogen to form NADH. Same thing down here. We're forming FAD, FADH2. Now FAD is picking up 2 hydrogens, whereas NADH only picks up 1. And then we're also producing 2 ATP molecules as a result of the Krebs cycle. Alright, so essentially the 2 main products that you need to understand that are formed in the Krebs cycle is NAD and FAD, and the purpose of those 2 energy carrying molecules is that they pick up and carry electrons and hydrogens that are needed in the electron transport chain to make ATP. Okay, so now we're going to talk about the 4th stage of the of cell respiration, which is the electron transport chain. And so the location of the electron transport chain is the inner membrane of the mitochondria. We call it the cristae. Now this is where we produce a whole lot of ATP. Essentially it's approximately 30, it's 32 to 34 if you account for glycolysis ATP production and the Krebs cycle ATP production. But essentially this is a high energy production process or stage in cell respiration. The reactants needed are NAD and FAD, okay, and remember that NAD and FAD were produced in the Krebs cycle. They carry these high energy electrons, okay, and they also carry these hydrogens, which are going to be used as protons, and they're going to be pumped across the membrane, and they're going to be used to go through the ATP synthase to make ATP. And then also we're releasing oxygen, okay, we're inputting oxygen here. The products here, we're producing about 32 to 34 molecules of ATP, so high production of ATP molecules right there. And then when NAD and FAD are all used up, okay, when they're oxidized, they're going to go back to NAD+ and FAD. They're going to go back into the Krebs cycle to pick up those electrons and hydrogens again. And then also we see the production of a water molecule. Okay, so what actually is occurring in the electron transport chain? So NAD and FAD are going to donate their electrons to the electron transport chain. FADH2 donates its electrons farther down, and I'll show you a diagram in a minute. Electrons are going to move down the electron transport chain just like in photosynthesis, and as those electrons move down the electron transport chain, hydrogens are going to move across the membrane into the inner membrane space. And that's so important because we want to we want a high concentration of hydrogen ions in the inner membrane space so that they can move through ATP synthase to make ATP. At the end, an oxygen is going to pick up electrons to form a water molecule. Okay, the hydrogens are going to re-enter the matrix through ATP synthase, and then the ATP synthase is going to spin and convert ADP into ATP, and we're going to produce a whole lot of ATP here. So now what we're going to do on this slide is we're going to go over and demonstrate the actual process of the electron transport chain of cell respiration. So remember, in the Krebs cycle, 2 energy carrying molecules are produced, NAD and FAD. So imagine NAD and FAD are coming in to the matrix of the mitochondria, okay, and then they're going to show up to the inner membrane of the mitochondria along the electron transport chain right here. And remember, NAD and FAD carry 2 very important things. They carry hydrogens, okay, which are going to move across the membrane into the inner membrane space, and they also carry electrons, which are going to travel down the electron transport chain, and they're going to pump hydrogens across the gradient. So we're going to start with NAD and FAD. So NAD and FAD are going to enter the matrix, okay, and what they're going to do is they're going to break off these hydrogens, and as those hydrogens are broken off, electrons are going to be released. Once those electrons are released, they're going to go into the inner membrane of the mitochondria, and those electrons are going to travel down the electron transport chain. As those electrons travel down the electron transport chain, hydrogens are going to move across into the inner membrane space. And then again, the reason why we want hydrogens in the inner membrane space is so that hydrogens can move through ATP synthase and convert ADP into ATP by adding a phosphate to ADP. So essentially that is the electron transport chain. NAD and FAD, okay, come into the matrix of the mitochondria. They're gonna break the bonds in between the NAD section and the hydrogen. Okay, electrons are going to be released. The hydrogen is going to be produced in the matrix. Those hydrogens are going to move across as electrons move down the electron transport chain. We build a high concentration of hydrogens in the inner membrane space so that the hydrogens can move through ATP synthase to make ATP. How is that done? ADP is converted into ATP. Also, as these electrons move down, okay, these hydrogens are going to pick up oxygens to form H2O as well. So that's again another byproduct of cell respiration is water. Okay, so that is the electron transport chain. And remember, the whole purpose of the electron transport chain is to accept NADH and FADH2, use their electrons and hydrogens, okay, to essentially make ATP and a whole lot of ATP. So that is the electron transport chain, and again, the whole purpose is to make a lot of ATP. Now let's review our diagram one more time and conclude it all. So remember, the first stage of cell respiration is glycolysis. We're splitting one glucose molecule into two molecules of pyruvate. We're also producing two ATP molecules and an electron carrying molecule, NAD. Pyruvate is then going to enter the mitochondria. Okay, pyruvate is going to bond with coenzyme A to form acetyl-CoA. Remember, pyruvate cannot enter the Krebs cycle. Acetyl-CoA can, because it has this acetyl group which initiates the process. So pyruvate is going to bond with coenzyme A to form acetyl-CoA. As a result, a carboxyl group is lost to form carbon dioxide. So again, that is carbon dioxide is being released in this process, and that's what we exhale out of our mouths. Now, acetyl-CoA is going to enter the Krebs cycle. Okay, it's going to be a series of energy extracting reactions, and essentially what we're doing in the Krebs cycle is we're breaking down acetyl-CoA and we're producing NAD and FAD, which carry electrons and hydrogens needed for the electron transport chain. Also, what we're producing in the Krebs cycle is two ATP molecules, okay, as well as carbon dioxide. So, after the Krebs cycle has produced NAD and FAD, they are going to be carried over to the electron transport chain, okay, and remember, the hydrogens and electrons are going to be dropped off at the matrix, okay, of the mitochondria. The electrons are going to travel down the electron transport chain. And as those electrons travel down the electron transport chain, hydrogens are going to be pumped into the inner membrane space of the mitochondria, which is called essentially the cristae. Hydrogens are then going to be we're going to have a high concentration of hydrogen ions in the inner membrane of the mitochondria. Okay. And then those hydrogens are then going to be used in the ATP synthase to convert ADP into ATP, to make a whole lot of ATP, and essentially it's going to be 32 to 34 molecules of ATP. Also in the process here in the electron transport chain, those electrons moving down the electron transport chain are going to be accepted by hydrogen, and then oxygen is going to pick up those hydrogens to form H2O, which again is another byproduct. Alright, so that is the whole process of cell respiration. Okay, remember, we are netting a whole lot of ATP molecules. In total, if you add up glycolysis, the Krebs cycle, and the electron transport chain, we get and make about 38 ATP molecules per one glucose molecule. That is a whole lot of ATP molecules, and that is essentially what is fueling our cells and allows us to function during our everyday activities. Okay, that concludes our notes on cell respiration. And remember, the process of cell respiration is where we convert chemical energy in the form of glucose into cellular energy in the form of ATP to fuel our cells.